Another one, let's talk about biocontrol just for a bit. Biological control and succeeding with biopesticides. Yeah, how to do this. What is biological control of plant diseases versus insects? You might have heard of like insect management by conservation and refugia. Unfortunately, unless the pathogen itself is an actual animal, such as this nematode, it's a type of animal, like an insect, there are not a lot of natural enemies. This pathogen causes tomato and tobacco ring spot of blueberries and amongst other types like this nasty Ziphinema. Yeah, refugia. Unfortunately, while they're great for sources of natural predators of insect pests and other types of things, they're just, and unmanaged crops are just a haven for pathogens such as this beautiful currant planting. You're like, where are the currant plants? And amongst all the goldenrod and other weeds. Nope, they're just refugia. And not to mention, refugia around apple orchards can sometimes lead to things that look like this. The fly speck sooty blotch over everything when you thought the apples were clean, and then you put them into a post-harvest situation. What else is a little different? Well, most of the diseases are caused by fungi and bacteria, such as this beautiful fire blight and that beautiful, like bitter rot of apples caused by Colototricum. There's no attraction to the host whatsoever. It's just particle physics, meteorology, and biochemistry. So these particles of bacterial cells and fungal spores, they're just moving together according to the principles of meteorology and sort of biochemistry, host pathogen interactions. And so in that sense, you're not going to be trapping for them. You can to kind of know when the meteorology is working, but for the most part, we'll forecast. And this has become a better way on how to be successful with it. This is showing some forecasting model outputs for fire blight, model out of Washington State, model developed out of Maryland, using arm farm data and predicting risk of when things are going to be there. You're literally forecasting like forecasting the weather. Do updates on this 1 to 2 times a week. The other thing to think about too is that most of the pathogens that we're dealing with in apple are actually not invasives. They tend to be part of the natural microbiome and ecological balance, except for a few, such as this gross citrus canker picture there. And what ends up happening is by the opportunity of planting a orchard, we disrupt the natural world. You saw what the refugia looked like, just weeds and plants and things everywhere. And we put down a very ordered apple planting in the area. And once that balance is disrupted, the pathogens are like, oh, hold on, we can try to bring things back to order and sort of get this plant that is taken over and manage it back down. So we sort of disrupt the ecosystem by growing food crops, and then this sort of favors pathogen population outbreak, which leads to what we believe is a disease and a problem, but it's really there to kind of keep host plant populations in check, such as this beautiful fire blight. I cleared out some area and planted some apples, and fire blight is like, hey, hold on now, you're out of control with the natural order. Let's sort of whack this back into situation, if you will. Keep it in check. So in terms of the insect world, they often talk a little bit about a couple different procedures for biological control. Classical, where you import an invasive and establish it. Like I mentioned before, there are few natural enemies of microbes, and this seems to be the case and with medicine as well. We're not really going to be able to bring in natural enemies of bacterial spotted stone fruit, other phloem limited type materials, either viruses or bacteria, such as in peach, or mycoplasma, such as in phytoplasma, such as in peach X disease, and rosette disease types of things as well. So we're not going to really import in. We will, but we'll talk about it in a different context. We're not really going to import in a lot of natural enemies. You don't only see this in medicine. It's not like we're going to bring in a new species of bacterium from another cow and eat it to cure our diseases. Or we're really not going to be able to facilitate the establishment of existing natural enemies. You can, but it just means pushing up the peach orchard in that particular instance. We won't really, having a lot of weeds and things like that is just going to lend to issues, either vectors or other stuff. And I mentioned before, the weeds and refugia are going to host pathogens. And in this particular instance, we darn a lot of agricultural practices that favor enemies and then end up leading to things. Like, for example, if I leave, this is the mummyberry disease and a blueberry orchard, and leave all the moss and the thick weeds down there, it makes a perfect environment for the mummyberries to make their apothecia, which inject spores and cause trouble. So we want to manage them. We want to bare ground, clean out, so there are not a lot of natural enemies. But what can we do? We usually do a process called augmentation. We sort of take a naturally derived agent. It's kind of invasive, but we put it back into the ecosystem. The nice thing about it is that it's not very good at being an invasive and doesn't get established. We would like it to get established because then it would eliminate things like apple scab. And one of the things to note is that limited optimization is allowed. We could take this Strobilurus tenacellus and sort of smush it and smear it, make a tea with it, and spray it on apples. Or we might take this wonderful RVI6 apple and plant it. That's perfectly fine as well. Or I could take up this Streptomyces from a manure pile and sort of make a soup out of it and spray it on apples, and it's okay. It's still biological control. However, if you take these same things, Strobilurus tenacellus, actually what it does is it makes a compound strobilurin A. If we make the soup, we can still use this, and it will bind to the electron transport chains. Back to that high school biology, it's basically cyanide for fungi only, not necessarily people. But if a chemist, let's say BASF, makes this, reduces the persistence of it, and reduces non-target effects, and then it becomes a different molecule, paraxylstrobin, and it is no longer biological control. Malus floribunda has RVI6 for apple scab resistance, and we can breed to move this thing around, but if I were to take it and put it into a Macintosh apple, and then it becomes a plant-incorporated protectant. It's still classified as biological control, but we're not going to be using it for organic agriculture. If I find an actinomycetes from a manure field and it happens to be fermenting under natural conditions, this secondary metabolite that will bind to the ribosomal protein S12 and kill off all my fire blight, I can make a poop soup with it, it's fine. If we have a chemist synthesize that and be like, oh, that's streptomycin, then it's become something we really don't want to use in the biological world. And what are these various biological controls? We usually refer to them as biopesticides, a couple others, minimum risk fungicides, and generally regarded as safe products. The technical definition of a biopesticide is a naturally occurring derived pesticidal substances. All of those soups of Strobilurus and cow manure, actinomyces would be just fine. Plant extracts, minerals, microorganisms and their products, and then, of course, plant-incorporated protectants. Now, they vary a lot in terms of type, cost, efficacy, but they're generally low risk for resistance. If you saw part one, you get it. They are hitting multiple different modes of actions, with the exception of a few, like the RVI6, which is going for a single gene, and the strobilurin A, which will be just targeting the mitochondria and the ribosomal transport chains. You can find the list of the EPA definitions of biopesticides right there on their website. You can kind of take a look. It will actually tell you the same thing that I just already mentioned. Naturally occurring, oh look, there they all are. It's a little bit different. And then the FDA has generally regarded as safe food additives as well. We can take a look at their page. You can read about it. You can read the notice. You can check the GRAS inventory for human food and what types of things as well, and whether they can be used for. And then, of course, EPA has minimum risk pesticides. They're so innocuous that they're generally exempt. You can take a look at their stuff as well. Oh, wait, hold on. I must have botched it. Let's try this one. No, it's gone. I love moving it around. Oh, well. Let's see if we can find it by just cut off the end of it there. Here they all are. Yep, move that out there. Just got to update that link and we're good to go. And you can read all about what are minimum risk pesticides, how they enforce it, conditions, what is a pesticide, etc. Let's see if we can find out anything about them. Looking for them. Learn more about minimum risk pesticide. There's the page I was looking for. Criteria, and you can view the list of active ingredients here. Pretty cool stuff. Final rule. Let's see if we can find exemptions. Let's see. I want to see if we can find the actual inventory list. I'm going to view the list. Where is the list? did find the list. Of course, now that we're doing this live, I will never find the list. Oh, I'm sure it's interesting. I don't see the list. How to use this list? More information on the rules. More information. Well, not finding the list yet. Oh, interesting. No, those are just the code of regulations. I wonder where my list went. Oh, no! Go away. Well, it seems to be a rather circular argument. Anyway, we'll find it next time. But what else do we know about them? How do they work? A couple different ways. Modes of actions. You've got some fun things here. You got fire blight of apples. You got powdery mildew on apple, and you got fly speck sooty blotch, and you got rust. Good things that can be used. Maybe not rust, but the others can be somewhat managed with the biologicals. They can do two things. They can kill microbes by producing metabolites. They can use competitive inhibition. You put these down in the floral cup, and you keep things from, like fire blight, from getting into the floral cup and causing disease. Usually these competitive inhibition things work best in flowers. Clog the hole with another organism, and the pathogen can't get through. And then finally, they can slow infection defense by boosting plant defenses. Some of the things that we thought worked by competitive inhibition may also boost on plant defenses as well. Mentioned before, most are just protectants, eradicants. They're going to protect against new infections or eradicate pathogens on the surface. They're not going to have limited persistence. You mean you'll have to apply them often, like almost as much as twice a week if it's raining a lot. And you might hear of some. A lot of these are the bacterial soup materials that have been fermented to produce antimicrobials, double nickel serenade, Serafel Tegro. Blossom Protect is something that was believed to do a little bit of competitive inhibition, but may turn on some defenses as well. Other lupin extracts, stylet oil, essential oils, naturally produced antibiotics can count as well. And then we have the defense inducers as well. As we mentioned before, Blossom Protect might sort of fall into this category now based on new science. And they will work internally. They turn on plant defenses and other defense processes in the plant, and they can be natural or synthetic, such as Actigard versus Lifeguard Regalia and employ Invasiplant. And even things as PGRs can sometimes enhance these sort of processes. So why would you want to use one? Biopesticides generally have better consumer appearance, acceptance, and appeal. You might be growing organic. You have an operation that protects against disease. In this case, I would like to think covered production operations. Or if you had tunnels, or if you're able to put these apples under tunnels, most of the cases for strawberries. If your operation is small and you can spray much, I can spray twice a week, and the disease pressure is light, maybe because you are using something that protects against disease. And it could include cover, it could even include using disease resistance. And then, of course, it's got to be the right pathogen. Something that's unprotected on the surface and fragile. The fire blight bacterium is just right there in the floral cup. It's oozing on the shoots. It's outside. You want to actually get it before it looks like this. It's right there multiplying on the stigmatic surface of flowers. Kill it. A powdery mildew, burn that right out. It's right there in the top couple of cell layers. That fuzz, that's actual powdery mildew spores. What else is good? Powdery mildews are good. Gray mold is good. Botrytis. It doesn't matter if it's a strawberry or an apple. Leaf blight, that fungus might be entrenched a little bit. If it gets too out of control, you're not going to be able to stop this with gray mold. So it's both good and bad. Powdery mildews generally work pretty well with biologicals. If it's a virus, nope, that's inside the plant. You'll notice that for your own human medicine, you don't do a lot of things to... you don't take a lot of antimicrobials or things for viruses. They're very limited antivirals. Botrytis shoot blight gets too deep inside the tissue, forget about it. Mummy berry? Well, if it's infecting the flowers, maybe. Anything that disturbs these cups, possibly. Red steel, this is Phytophthora infection of strawberry. And it's right there inside the plant. Is there anything that gets inside the plant? Yep, there are a few things, but not many, so it might not always be the best bet. Could you dip your plant, strawberries, in a soup of an antimicrobial to protect them from getting infected from the outset? Possibly. Yeah, so that's sort of a question mark. It really depends on the situation. How to beat them? Use the best horticultural practices. The high-density apples, in the case of the apple order, are the best. Better for color, yield, agrochemical applications, drying time, air circulation. Just imagine all the better color, air circulation, and drying in here versus the sort of tall spindle, semi-dwarf plantings of old. This is just accumulating a microclimate that's conducive of disease. The third thing to do in order to be successful is use disease forecasting tools. They're going to predict the conditions that the pathogens mature, dispersing, and infection. This will increase your precision and increase the chance of getting a biopesticide to work. Now, climate change can lead to erratic weather, which may force you to be more agile and may cause the models to behave a little more inconsistently, but they're still better in terms of precision and effectiveness than just picking every Monday, etc. We have the newest system. I talk about it a lot, and it uses both local and satellite weather data. You can pick neighboring stations to look at it. We see this quite often. I need to go back in and reset the browser magnification back up to 100. On this place there, you can put all of your different materials in there. It looks like I signed in on Mills River in North Carolina to watch that station, as well as part of my research. Well, that's it for biopesticides at the moment. I hope you enjoyed the video. See you in a little bit.